Today, we are going to break down some absolutely mind-blowing research that could literally change the game for treating brain and spinal cord injuries. We're talking about a cancer drug that might just unlock our body's secret ability to heal its most important wiring. Let's get right into it.
So let's start with the question that has honestly stumped doctors for, well, centuries. You get a cut, it scabs over and heals. You break a bone, it mends. But the central nervous system, your brain and spinal cord, that's a different story. Why is it that when those get seriously injured, the damage is usually permanent?
To really get it, you got to think of your nervous system as this massive, intricate network of superfine biological wires. These are your nerve fibers, or axons, and they're the communication lines carrying every single signal between your brain and your body. An injury here, it's like someone just went and snipped a critical fiber optic cable. Now here's the problem. Unlike a nerve in, say, your finger, which can try to regrow, a severed axon in your brain or spine does the opposite. It basically panics, seals itself off, and forms this thing called a retraction bulb. It's a dead end. The nerve literally just pulls back, puts a cap on itself, and stops all regrowth cold. It's the biological version of just giving up. But what if you could tell that nerve fiber, hey, don't give up? Well, a brand new study has found a really surprising clue. And it was hiding in a place you'd never think to look, an FDA-approved cancer drug.
The drug is called Epothilone B, or EpoB for short. So its day job is fighting cancer, and it does that by stabilizing a cell's internal skeleton, these tiny things called microtubules. It basically locks them in place so cancer cells can't divide. But it turns out that this exact same stabilizing power does something completely remarkable when you use it on an injured neuron.
And I mean, the results were just stunning. In the lab, without any treatment, only about 16% of severed axons even managed to stop degenerating. But get this, when they added EpoB right after the injury, a whopping 90% of them either stopped dying back or, and this is the key, they actually started regenerating. That's not just a small improvement, it's a total game changer.
Okay, so when you see a result like that, the next question is obvious. How? What is this drug actually doing inside the cell to flip that switch from give up to let's rebuild? What does that process even look like on a molecular level? To figure that out, the researchers had to see inside the healing process with a level of detail that just wasn't possible before. They needed some seriously high-tech imaging to watch the cellular machinery get to work almost atom by atom. So they used this incredible technique called cryo-electron microscopy. The best way to think about it is like this. They flash freeze the nerve cell at different moments while it's trying to repair itself. Then they use a super powerful microscope to build a 3D movie of everything happening inside. It's literally like having a molecular camera filming a construction site. And with this camera rolling, they could finally see what was happening right at the tip of that injured nerve. And what they saw, well, it completely threw out the old textbook ideas about how nerve healing is supposed to get started. For decades, scientists thought that nerve regrowth started with this delicate little structure called a growth cone. The idea was that this cone would kind of gently feel its way forward like a little hand searching for a path in the dark. But when they looked at the axons treated with EpoB, that gentle little growth cone was nowhere to be found, at least not at the beginning. Yeah, instead of a gentle search, what they saw was pure brute force. The axon wasn't feeling its way out, it was ramming its way forward, pushing with incredible power from the inside out. And this right here is the central secret that Epo unlocks. That force wasn't just random chaos, it was organized, it was directed, and it was powered by the cell's own internal highway system. Now pushing force comes from those microtubules we talked about. They're the cell's internal skeleton. Think of them as both the structural scaffolding and the railroad tracks that transport all the essential cargo up and down the nerve. Normally, after an injury like this, those tracks just fall apart. But this is EpoB's magic trick. It acts like a super stabilizer for these microtubule tracks, refusing to let them collapse. Instead, they organize themselves into these powerful, growing bundles, and they just keep growing, extending with so much force that they physically push against that sealed-off tip of the nerve. Imagine what that does. All that constant pushing creates insane tension on the outer membrane at the nerve's tip. It's like blowing up a balloon and stretching it right to its breaking point. This high-tension state is a critical step. It's like a screaming emergency signal to the rest of the cell, saying, hey, we need building supplies at the front line now. So that leads to a new problem.
right? If you're going to build this fast, you need a ton of building materials, and you need them on-site immediately. Where are all the new pieces for these microtubule highways coming from?
Well, it turns out the cell has a genius logistics system. It doesn't waste time making new bricks from scratch right at the construction zone. No, it does something way smarter. It ships in prefabricated parts ready to go.
These ready-to-use parts are called tubulin clusters. You can basically think of them as coiled up, ready-to-install sections of new microtubule track. The cell ships these coils down the existing tracks to the injury, where they're rapidly unspooled and just snapped onto the growing ends. It's an incredible piece of cellular engineering.
Okay, so let's put this all together. With all this new information, the researchers could actually map out the entire recovery process. They created a clear 4-phase blueprint of how this drug-powered healing actually works.
So it all starts with phase 1. The injury happens, and the nerve seals itself off into that dead-end bulb. Then comes phase 2, the real game changer. ApoB stabilizes those microtubules, and they start that forceful push, creating all that tension. That tension triggers phase 3. The cell rushes in new materials, the tension gets relieved, and a proper new growth cone is finally built. And that leads to phase 4. The new growth cone takes charge, and normal, steady regrowth can finally get underway.
What this all means is that for the first time, we have a detailed molecular roadmap of what a successful nerve regeneration looks like. We're learning how to turn the cell's own powerful but usually silent repair machinery back on. This isn't just an interesting theory anymore, it's a visual guide for how to rebuild a nerve.
And that just opens up such a fascinating door, doesn't it? We now have a step-by-step blueprint for kickstarting healing where we once thought it was impossible. It really makes you wonder, if we can master the art of rebuilding a single one of these biological wires, what's to stop us from one day learning how to rewire the entire nervous system? That is a huge question, and the answer could absolutely define the future of medicine.